I'm Hwan Chang. Today I'd like to present a reconstruction of the gene-regulatory network based on the repairing sparse rank matrix. This is joint work with Roll, Palak, and Dr. Joe Gray, and then Professor Claire Tomlin. Recently, many researchers use the high-throughput measurement data to construct models of complex systems such as breast cancer signaling pathway. And we consider time series gene expression under various perturbations using the small molecule inhibitor. So you can see that in the here, lapatinib and AKT inhibitor represent the small molecule inhibitor that affect our system. And even some mutation cases, such as PI3 kinase mutation, you can imagine that there's only single influence change with the wild type expression. So if you consider this time series gene expression data set under various perturbations, and there might be many problems. First, we only have the partial information about this perturbation. For example, when you apply the AKT inhibitor, you know that the expression of the AKT will be decreased, but you don't know the exact dynamic of this perturbation. Due to this property, if we use this data set for the infer the graph structure, and this perturbation can distort the relationship between genes, so you may mislead the inference. And also many graph inference problem or algorithm apply to the each data set independently, so there is no use of a commonality. So question is, how do we incorporate all the data set we have? And then what extent do we incorporate all the data set? And how do we handle this partially known perturbation information? So our goal is that correctly repair or extract the common graph structure across the perturbed gene-regulatory network structure. So in computer vision application, so-called image impairing or image completion technique can repair this corrupted image like this. So the idea is that the underlying original image shows the near regular or regular pattern, so that the original image can be represented as original image has the row dimension in this problem. So the recent paper used this sparse row rank texture idea so that they can able to infer this corruption and then repair the original image. So motivated by this, we consider this using this older data set to infer the common graph structure. So this is kind of a conceptual diagram with a metaphor in this computer vision application. So you can imagine this time series gene expression data set with various perturbation as a corrupted image in this in figure A. And once we can infer this direct perturbation from the data set, and we are able to correctly repair the common graph structure in C. And this is the overall procedure. So since we have the various experimental data set on the various perturbation, and you can see that each perturbation is partially perturb our system. So once you construct the sparse row rank matrix representation that I'll show in a few slides, and then you can imagine that each data set is partially corrupted, so you can use the other data set to repair this part, so that you finally can repair the original common graph structure from this perturbed data set. So in the classical way, the biochemical reaction can be modeled as follows. So you can imagine that x dot equal fx plus u, and u is the direct perturbation. And fx can be represented by this q multiplied fb. So q represents either parameter or the topology of the gene-regulatory network with this fb, that is the candidate basis function you can plug in your model. So you can include a linear function or nonlinear function such as Hill function or quadratic term in your dictionary. And we basically reformulate this original equation into this formulation. So we vectorize this Q into the single vector S, and then we define this set is a phi, and we have the U. Since this original structure Q is sparse, so that this vectorizer of the Q is also sparse. And since we have the time series data set at discrete time point, we have the YTJ and the VTJ and UTJ and the SJ corresponding to the this structure of the at time TJ. So in many cases in the graph inference problem, U is assumed to be known, so that you just subtract the U from the Y, and then you basically find the S, penalizing with the sparsity of the S. However, here we only use the partial information about U. In practice, you don't know the exact amount of the U. So here we only assume that if you put any stimulation or inhibitor that affect your system for the case gene, and we only know that UK is non-zero, and the other part is 0. So we use the uncorrupted information to repair the graph structure. So in the previous slide, I present where the sparsity comes from, and then in this slide, I showed why we are looking for the row rank property. So remember, SI represent the graph structure, so we can stack in a columnize of all the different SI over time, and then we can also stack all the S, this capital S across the different perturbation, like different experiment setup. So we have this kind of calligraph Q can be represented by a sparse and row rank matrix. So you can imagine that this simple topology can be represented by this calligraph Q matrix. So sparsity comes from the characteristic of the gene regulatory network, and row rank property comes from the commonality across the time versus the different experiment setup. So we basically looking for this optimization problem. So we want to find the sparse and row rank matrix of the Q, and also we penalize the U because the small molecular inhibitor only affect a few gene expression, and subject to this relationship that we derived in the previous slide. Here's the P omega is a linear operator that restricts the equality to the element in set omega. And this set omega can be defined as follows. Since we know that the experimental condition, we know that we can use that information with this defining this support omega. And we can basically use the uncorrupted part of the experimental data set to reconstruct the original graph structure. So we can estimate the corrupted signal UI and then redefine the set omega iteratively until converge this algorithm. So the idea is that we refine the support estimation, and then we are able to repair the original Q. So here's a simple example. We randomly generate the graph structure, and then we simulate with a known dictionary. So we use this input perturbation, and then you can also represent this graph structure into this calligraph Q. So here, red represents the activation, and then green represents the inhibition. And basically, this in figure A, we represent the calligraph Q that we defined in the previous slide. So here, red represents the corresponding the stimulation edge, and then green represents the inhibition edge, and then this black color represents the zero component. And this is the true input that we generate this data set. So the proposed method able to repairing the common graph structure in A. And this is quite consistent with the true graph structure. And we also applied the L1 optimization here, and then here each data set is used independently. Since we don't know the exact information about this perturbation, all the inferred graph structure could be biased, and even they are not consistent across the different experiment setup. And also we start with this initial mask because we only know that a certain part is corrupted or not. So we start with this binary input, but finally we converge to this estimate input, which is consistent with the true input. And since the previous example, we generate the data set, so we know the exact dictionary. So here we apply the Dream4 in silico network challenge data set. So this data set has the time course under various perturbation and generated by the biological plausible in silico network. So here we consider 10 nodes, and then this the The red color represents the activation, and then blue color represents the inhibition. And this is the experimental condition in this DREAM4 data set. So here, for example, experiment 3, we know that G1 and G2 and G8 is perturbed by the drug. So we only use that kind of information to repair the common graph structure. So this is the plot of all the raw data set. So here we have the time series expression of the G1 to the G10, and different color represent the different experimental condition. And here you can imagine that this data set we can separate this raw data set as a perturbed and unperturbed response by using the experimental condition. For example, this gray color represents the directly perturbed response from this raw data set, and it's corresponding to this corrupted image. And non-gray color represents the unperturbed response, so that we want to use that information. And you can imagine that this non-gray color represents this clean image in B. So you basically use this information to repair the entire structure. So this is the inferred graph structure we have. And so here the solid line represents the exact match with the ground truth. And here the line represents we miss. And then the dashed line represents we have the false positive link here. And we can also estimate how this drug perturbation affect our system by extracting this common graph structure. And we want to further analyze this result with the expression data set. So there are many implications. So first, if the data set reflects the dominant common dynamic responses, we are able to infer corresponding edge. And it turns out in some cases, we actually avoid overfitting. So I show one example here. So I just consider this part. So here I plot all the raw data set. So x-axis represents the G1, and then y-axis represents the G2 versus G2 to the G4. So in C and D, the time series gene expression shows the common dynamic responses, so we are able to infer the corresponding edge. And in figure A, it's a plot of the G1 versus G2, and we know that this experimental 3 is directly perturbed by a drug, so we remove this part. And the other data set shows that we cannot there's no way to infer this part, because here, this is time series gene expression data set, but although G1 is changing over time, but G2 is never changing. So this data set doesn't reflect this inhibition link. So if you infer this edge from this data set, you have to use this direct perturb information, which is unknown. And in B, there's two dominant responses corresponding to this inhibition, so we are able to infer that. And here we only have one data set corresponding to this part, so we missed this link. So just to wrap up, the proposed method can extract the common graph structure by using the rank property, which penalizes the commonality across the experimental setup. And we can estimate the unknown drug effect by separating the common dynamic response from the perturbed gene regulatory network. And from the lesson from the DREAM4 data set, the proposed method informs us how to design the experiment. So we should perturb our gene uniformly well. The intuition is that if you remember the computer graphic application, if you block the entire row, and then there's no way to repair a corresponding part. So that we have to uniformly perturb our underlying system in order to correctly repair the common graph structure. So we are currently applying this method to the large-scale data set and using this tool to design the effective experiment to infer the HER2 breast cancer signaling pathway. So thank you for your attention, and I'm happy to take any questions and comments.
So there is time for one or two questions.
I have a question about your low rank assumption. So basically, you're assuming that the observed networks under different perturbations is the sum of a low-rank network, which is a common subgraph, plus some sparse noise, which comes from the perturbation.
Yeah.
But why do you have this assumption? How do you know that this common graph is low rank? Do you assume that there are like some factors that driving the expressions that you see, and because of that you have low? Because the argument that you gave about this assumption also relates to the perturbation, but that's the assumption that you have for the sparsity, not for the sparsity of the spectrum, which is low rank. And the second question is that even if you have this assumption, how do you make sure that this is the lowest rank common subgraph, common graph that you infer? Thank you.
Yeah. So that's a great question. So the sparsity comes from the characteristic of the generator network. And since we assume that the older system has the same structure that we formulate into this like calligraph Q matrix, so that they have to be consistent across this columns, so that the row rank comes from that part. And also
But that's the sparsity. Like you have a network.
No, sparsity comes from the vectorized S, and then we stack all the S over time across the experiment.
That's time, right? That's time sparsity. But how that is related to the spectrum sparsity of your network? Spectrum comes from the sparsity of the generator network structure itself, not the structure of the Q, right? Okay.
So you can imagine that the parameter doesn't change over time if you build the ODE model. So that can be represented by the multiplication of the S multiplied one vector. So that has the row rank, right? So we basically stack all the different experiments over time so that the original calligraph Q matrix should be represented by the row rank. And also in this paper, we actually, if you consider like large number of candidate basis function, this row rank property might not be able to repair the common graph structure. So instead of use of the nuclear norm, we use the total variation if you consider a large number of candidate basis function. And then sparsity of the U comes from the experimental design, because the small molecule inhibitor only affect a few genes. So that comes from. Is that answer for your question? We can discuss offline. Yeah.